I want to talk a little bit about data management and tracking, which we covered slightly here. But I want to talk a little bit about data collection, how it's collected, what are the best systems solutions for doing so, setting up the right data models and infrastructure, what have you. Omkar, I'd like to ask that question of you first.
Yeah, absolutely. So your question has so many parts to it. Right? Let me piecemeal it again. First questions first. And I think one of the audience members also asked this question, and actually it's very applicable because if you see the mix of the audience, more than 50% are early stage in development, right? So when do you start thinking about this? I would say now. Start doing it now, because now is when you start collecting that data. These might be difficult conversations to, for your leadership to think about, and put me in front of them, and I'd be happy to talk to them about it. But the thing is, the more data you have, the more data you collect, the more data you feed into whatever analytical algorithm you build, the better is the more reliable is the outcome, right? So that's the question about when. Second part of your question, Erin, was the how to collect the data. That data at this point sits in disparate systems, in various systems. Some of it is in paper form, some of it is structured, some of it is not structured. And more importantly, in earlier stages of development, you are still working with a very defined set of material, right? Like what type of donor cells you're getting. And this has happened in the allo space, especially where we identified a specific genetic marker in a donor that makes it less probable for LV tropism, but it was much later in your stage of development. So the more you're able to collect this real world data through various means, right? You can do it through systems based integrations, through manual entries or paper sources, but you generate that body of evidence over a period of time, the historical data set. And as I said, the more batch numbers you have and the more batch numbers increase over a period of time, the larger the data set, the larger the pool of data you can start to mine and extract trends and information, useful insights from. And the better it will help you inform your clinical protocol, your manufacturing procedures, your starting material collection parameters, out of spec criteria, etc. So I can give you, again, I don't want to take up too much of the time over here, but I can give you a very specific example if you were to dig deep into how a digital solution or data analytics has been able to address variability, right? One of the partners in the firm that I work with, Lax Pernenkil, he's been working on something called as cognitive spark for manufacturing. What it is it's meant to ingest a lot of in-process data and provide actionable insights and recommendations through inbuilt algorithms, right? And so we actually tested this out as a pilot, not in the apheresis setting, but in the plasma collection setting. But you can draw a lot of parallels, right? The business question that we're trying to solve is, one, the operator does not have visibility into the yield of the batch until it's completed. Two, it is unclear which parameters or factors affect batch yield and by how much, and what can you do to actually reduce the impact of those anomalies. So as part of this pilot, what CSM was able to do is collect data from various different sources, identify the critical processing parameters, and perform either univariate or even multivariate analysis that allows you to proactively identify anomalous behavior. So in simple terms, it tells you what an in-control behavior of your CPP, your critical processing parameters, what those factors should look like versus what they shouldn't look like. And it displays a batch record sort of panel view to the operator. And to take it one step further, it also proposed immediate corrective actions for these at-risk parameters. So at the end of the run, you get like, oh, here are my top 5 positive contributors, here are my top 5 negative contributors. And if you were able to, again, have sufficient data across multiple apheresis or plasma collection runs, you are able to identify, if you do X, this is the impact. If you do Y, this is the impact, and to what degree that impact would be. So this is not rocket science. It is very much reality. It's just, again, me urging the industry, pushing the industry to actually put this into practice.